So without further ado, I am going to introduce Vic, who will introduce the speakers of this morning's session. Vic got his PhD at Johns Hopkins, an undergraduate at Caltech, his PhD at Johns Hopkins University, and then a postdoc at Penn State before coming here. He's in the Department of Biochemistry at the medical school, and a member of the Center for Advanced Biotechnology and Medicine, a faculty member here in the building in which this symposium is being held. Vic.
Okay, well, thanks, Paul. So I wanted to give a brief overview of the first theme. And as Paul mentioned, the ENIGMA project is divided into 3 categories. And the area that we're focusing on is really thinking about the very first protein catalysts, the first proteins that emerged from some sort of prebiotic primordial reaction and catalyzed the first reactions that were relevant to metabolism. And it's a real privilege to think about origins of life, but it's an even greater privilege to be funded to explore these sorts of scientific questions. And so I've been having a lot of fun over the past couple of years with the overall ENIGMA group. But I wanted to highlight in particular the team members in Theme 1 who have contributed to this. And thinking about early protein catalysts is not an easy task, and it requires a lot of different expertise. And so I'm not going to introduce everyone by name, but I just want to mention that this is the work of a lot of colleagues and collaborators who have contributed to this, and then also a number of postdocs who are highlighted in red, graduate students who are highlighted in blue, and then undergraduate students who are highlighted in green. And as part of our NASA project, we also have a training mission to take astrobiology and spread it through the scientific community. And one of the exciting things about astrobiology is because of its interdisciplinary nature, you really learn to think outside of the box and work in a lot of different areas. And I just want to point out a couple of our alumni who are now working in industry, Josh and Saroz and Doug. They're working in very different areas from astrobiology, but they're applying a lot of the same sort of understanding of protein modeling and protein experimentation that they developed with us. And then also postdocs who are now in academia, Hagai, Kenneth, who just got a job at Caldwell University here in New Jersey, and Banu, who's now a postdoc at Pullman with John Peters. They're all carrying on their research. And then we also have fantastic undergraduates who have come through this program. Shahir, who's now a medical student here at Robert Wood Johnson. And I want to mention Juliana, who this year is going to be joining the graduate program at Georgia Tech in biochemistry and chemistry, which is going to be doing a focus in astrobiology when she's there. So she's actually going to be one of the next generation astrobiologists coming out of this program. And so, as Paul mentioned, we're very interested in the very first proteins. And it's important to remember the assumptions, because there's proteins now do everything in biology. And so the question is, how do we focus on a set of proteins that are going to be doing interesting things, relevant things at the earliest stages of life? And so, we really are focusing on the oxidoreductases, enzymes that are involved in key reactions in metabolism. And this is because we think that the main driving force for life is redox disequilibrium, this electrical energy in the environment between electron sources and electron sinks that really drives all biological reactions. And today, we have a huge array of electron sources and sinks in the environment. And the same thing was true early on. And the question is, can we anticipate what those sources of energy were and how did metabolism take advantage of those? And so, if we focus on the oxidoreductases, then we really need to think about what are the core redox enzymes that, that essentially move electrons through metabolic pathways and take this energy. And so we find a set of enzymes, and then when we look at them, we realize that they are very complex. They're massive nanomachines. That's the N in ENIGMA. And so then our job is to identify not just the oxidoreductases, but what are the simpler forms that that led to these particular proteins. because we can't imagine a massive enzyme like this hydrogenase, for example, emerging from the primordial soup as it is. So part of the task then is to take these large proteins, break them into smaller blocks, which we call the Legos or the building blocks, of proteins, and see whether or not we can identify these essential domains. And then the last step is protein engineering, because we do not have any fossils of these proteins that we can go and look at directly. So we need a way to explore whether or not our hypothesized simpler pieces were real. And so that's the last step, where we actually take these larger proteins, break them into building blocks, and then make these blocks in the laboratory, see whether or not they have the essential function that we ascribe to them. And so this is something now we've been working on for some time. And we've gone through the protein database, which for those of you who are here in person will have an opportunity to see the protein data bank tomorrow afternoon. And essentially what this is a collective resource, a public resource that contains hundreds of thousands of high resolution structures of proteins. And what we were able to do was to find all of the oxidoreductases within that data set, look at their structures, break them into smaller parts, and then classify them, see whether or not there were certain parts, certain types of protein folds that were used again and again and again in metabolism. And then we built this network. And this network here describes not only the essential parts, each of these nodes represents one type of protein fold, but also the way then that they are connected together, the way that they are put together in proteins to assemble these larger machines. And so these are really electrical connections. This is one protein that is donating an electron to another protein, donating to another protein, and you see these wires going through the middles of these proteins. So this network as a whole is really a wiring diagram that shows us how electrons flow through metabolism. And this has been a tremendously useful resource over the past few years, and we use this really to guide a lot of our projects. And then one project in particular that we just submitted this year is finding one of these building blocks, a nickel binding site that has this essential property of reducing hydrogen, taking protons and converting them into hydrogen gas. And we were able to take proteins that are much larger, hundreds to thousands of amino acids, and identify the smallest component in them. And you're seeing here our model structure for a 13 amino acid peptide. So a really, really short molecule that is able to bind 2 nickel atoms. And you can see here, here's the coordination for this simple peptide. And this peptide has a lot of characteristics that are found in these much larger enzymes, but everything now is all the chemistry is happening in this small molecule. And what we find is that this small peptide is able to robustly produce hydrogen when it's provided with electrons. And this project, just like all the other ones in our theme, have required the expertise of a lot of different scientists, from spectroscopists to modelers to biochemists. And so it's been a large team that put this project together. And so this project's in review now, and hopefully it'll be published soon. Okay, so thinking now about where is theme 1 going? We have 1 more year, but we're thinking sort of broader than just the last year of ENIGMA. There's been a tremendous paradigm shift in structural biology just within the last couple of years. And I'm sure all of you are aware of this, but I'll just briefly mention that machine learning has done a lot to sort of increase our understanding of protein structure. And there's a competition in protein structure prediction where lots of different laboratories develop algorithms for predicting these 3-dimensional structure, the fold of a protein, from its amino acid sequence. And this has been going on for over 20 years. And in the last few years, programs that use machine learning to essentially predict the structure directly from sequence have started vastly outperforming all the other tools that physical chemists like myself have been developing. And what you're seeing here is the difference in essentially the precision with which you can predict the structure of protein compared to an experimentally solved structure. And you can see that a program like AlphaFold, for example, this dark blue bar over here, can predict the structure to within less than an angstrom precision. So an angstrom, for those of you who don't think about this, is less than the distance between 2 carbon atoms in benzene. That's about an angstrom and a half. So this is really, really high precision. And this is a program now that we can use to predict the structures of lots of different proteins from their sequence. Relevant to ENIGMA, not only can it predict the structures really well, but it can also predict the binding sites for metals. And one of the sort of the tenets of theme 1 in looking at the evolution of oxidoreductases is that electrons need to move through redox active cofactors within these proteins. They need to have essentially a wire with which they can move through the protein matrix. And these wires are essentially defined by the positions of metals and other redox active cofactors. And what you can see here is that while programs like AlphaFold and Rosetta don't actually put the metals themselves into the structures, because they've been trained on these large data sets like the PDB, they were able to accurately place, sorry, I keep losing the mouse. They're able to accurately place all the amino acids that bind these metals. So you're seeing here in blue, the predicted position of amino acids. And in green, their actual coordinates in an experimental structure. In the experimental structure, they're in those conformations because they're bound to this black zinc atom right here. But even though that zinc atom is not there in the prediction, the program knows to put those amino acids there, which now allows us to start looking for metal sites in proteins. We could see, for example, a group of amino acids that are just waiting for a metal to come in. So now we have the chance to take advantage of these tools. So how can we take advantage of them? Well, so one example is a project that is nearly completed now, where we're finding AlphaFold to be very helpful. We've been studying the evolution of ferredoxin, which is an electron shuttle. It moves electrons from donors to acceptors within the cell. It's a ubiquitous protein. It's found in all branches of life. And we're very interested in it because for a number of reasons, we think it's very old, and because its fold is very simple. So simple, in fact, that when you look at its three-dimensional structure, I'm showing you here sort of a side view of the molecule, and we color it so that the first half of the molecule is, let's say, yellow, is orange, and the second half is green. You can see by eye that there's actually an internal symmetry in this molecule. So this is a 60 amino acid protein, but if we were to cut it in half right here in the middle, we would now have 2 half proteins that if they were to come together, we could imagine it forming this ferredoxin shuttle. So potentially we could imagine a simpler ancestor to ferredoxin that was this, maybe just the green peptide or maybe just the orange peptide. And so we've tested this molecule in the laboratory, and in collaboration with our ENIGMA partners at Bryce, we've actually put these small peptides into cells and asked them to complement the deletion of a natural ferredoxin. So you take out the natural ferredoxin, put in our putative ferredoxin, and see whether or not it can bind metal, it can fold, and it can function within the cell. And what we found was that it did function, but only when you dialed the oxygen concentration in the chamber down very far. So you'd had to be at really, really low levels of oxygen. Then we were able to see the cells grow once we induced the expression of one of these peptides. And then we went back and we looked at the genomes of lots of different organisms, and we found that there are examples of these half ferredoxins, these smaller ferredoxins, in some organisms, but they're only found in anaerobes, telling us that if this was an ancestor, it was something that had to exist prior to the GOE, prior to the emergence of oxygen, or if it's in extant organisms, ones that live in anaerobic environments. So then Banu and Ian, 2 postdocs who were on the ENIGMA project, took one of these genes. This is just 130 amino acid half ferredoxin. They synthesized it. They showed that it could bind iron sulfur clusters. That's what this EPR spectrum shows you over here. And then we also did a redox experiment where we could put electrons on and take them off. And we found that this peptide, with its small size, was able to bind and release electrons just with the same energy, with the same potential as ferredoxin. But we didn't have a structure. So we took the sequence, and actually we took 2 copies of the sequence and fed it to AlphaFold in this particular case. And here's the structure that it returned. And you can see here, it looks very much like the full-length ferredoxin. And in fact, if you look closely, these little yellow groups right here, those are cysteine amino acids that are essentially pointing to a metal binding site. and waiting for an iron sulfur cluster to come. And of course, AlphaFold doesn't give me the iron sulfur cluster. But if I were to now take the experimental structure of a ferredoxin and overlay it on this model, and this is right here in blue, that's the model, you can see it overlays well, but more astonishing, you can actually see that the side chains themselves, here's the experimental cysteine, here's the model cysteine, they're within like a tenth of an angstrom of each other. They're essentially in the same place. So these programs will allow us to reconstruct the structures of these ancient molecules. And because we can take any sequence and give it to this tool and get a model structure for them, we now potentially have a way to go beyond the structures that are in the protein data bank. So this network that we made of electrical connections between folds, that was based on experimental structures that were available about 2 or 3 years ago. We went through the PDB and we built that. Now we have the opportunity to go through a larger database of sequences, ones that we don't know their structures. In some cases, we don't even know their functions. And alumni Adrian Horfrost with Jana Bromberg has developed a tool which can allow us to essentially predict the functions of these sequences and potentially identify new candidate oxidoreductases. And then we can take those candidate oxidoreductase sequences, predict their structures using AlphaFold or Rosetta. And then another tool developed by Ariel in Jana's group in Theme 2 has developed software for predicting the metal identities associated with these proteins, actually from sequence. We don't need the folds for those, but we can combine that information now to construct a model of the entire enzyme. And then we can use the same tools we've used before to break them into parts and potentially identify new components in this network that we were missing because the experimentalists hadn't looked there before, or because we didn't realize those proteins were oxidoreductases. And so we have the potential now to generate a complete span, which I'm calling the C-span. Hopefully that sticks. And that will now give us an entire sort of picture of the electron flow in metabolism. And so this is, again, this is a long project, but this really shows us now what this new structural information will allow us to do. Another thing that we're interested in is using tools like AlphaFold to start to extrapolate the lost structures, the ones that have disappeared over time, the transition fossils that are no longer found in modern organisms, but may have existed at some point during evolution. And we have a hypothesis in looking at this span network, that when you have 2 protein folds, let's say in this case, a Rossmann fold, this is a nucleotide binding fold that is found in a number of different metabolic pathways, and our ferredoxin fold that we've been studying for a while. We see that there are examples in proteins where the 2 of them are connected, where the 2 are found in the same protein. And so we have a hypothesis that when there's an electrical connection between folds, that may be evidence of an evolutionary connection, that at some point a ferredoxin was duplicated and you had 2 tandem ferredoxins, and then over time, one of them evolved or differentiated into a different fold. Now that differentiation process, that evolution process is gone. We can't see those intermediates, but could we potentially reconstruct them? And so this is a project that we've proposed with Professor Ben Tal, who's going to be talking later this morning, where we're going to use structure prediction tools like AlphaFold to take endpoints like the ferredoxin, and in this case, ferredoxin NADPH reductase, which has this Rossmann fold, and see if we can start to build these intermediates. And we can test them very quickly on the computer. A big bottleneck in these sorts of projects is solving the structures of these intermediates and trying to figure out whether they're viable or not. So this will sort of eliminate that bottleneck. We can rapidly explore lots of different evolutionary pathways between state A and state B, and then focus on those ones that seem to be the most promising. So this is very exciting. We can not only sort of complete the span based on extant members, but we can start to reconstruct the pathways and find these fossils that are lost to time. All right, and so then the last thing I want to mention, sort of thinking to the future, is that Right now, we've been very focused on identifying the parts, identifying a hydrogenase, identifying a ferredoxin. Potentially, you know, we're thinking now about identifying a nitrogenase, right? So we're thinking about these parts, but really, if we want to talk about metabolism, we have to think about the way they interact, how the electrons move from one molecule to another through an electron transfer pathway. And so there now we need to not just think about the individual components and how they evolved, but the individual components, but also how do they evolve? And so there's this emerging field of systems peptide chemistry, which our next speaker is going to be telling us a little bit more about, where we can not only think about molecules, for example, that have some function, but what is their evolvability? How did they come from smaller, simpler parts and develop these sorts of functions? And I think we need to learn from people who are already in the field of systems peptide chemistry, ways that we can start to think about this evolutionary process. So with that, I think I'm good to move on to introductions, unless there's any questions from the audience. Yes, or from the chat.
Yes. Is it a question from Bob Hazen? If alpha fold does not include the metals, could the presence of metals lead to a very different fold structure?
Yes, that's quite possible, right? And I think that, you know, there are proteins that we see this experimentally, that there are proteins that if you fold them in the laboratory in the absence of metal, they look like those alpha fold models. We call that pre-organization. They essentially pre-organize the binding site so that the metals can be incorporated. But there are also examples of switches. There are proteins that adopt one fold, and then when you add the metal, their fold switches to one that can bind the metal. And AlphaFold is really just doing pattern matching, right? It's trying to take the sequence and find a three-dimensional fold that matches that particular sequence. So if you have, for example, a fold that could switch between two different conformations, then it may be that AlphaFold can match it to both of those folds. And we may be seeing, you know, one which can bind to the metal and one which cannot. So the scenario that you're describing, Bob, is possible, right? But a lot of metalloproteins, and in fact, most ones that robustly bind metals that are not meant to be switches, they adopt a single fold, and often their binding sites will be pre-organized. So I hope that answers your question.
Yeah, thanks very much. I appreciate that.